So, hey everyone, welcome to Cognitive Science. So, as we chatted about a little bit last week, the aim of this class is really all about discussion. In fact, the less me and Brad yabber on, then the more you guys chit chat amongst yourselves, the better. And there's going to be plenty to discuss because there's probably not going to be. I would say there's definitely not going to be a single topic on which the world is on universal agreement, and that's just the nature of the field, and that's kind of what helps to make it fun and interesting, but also occasionally difficult and even frustrating. So, we can bounce ideas around, and who knows, maybe even this will be a venue in which people come up with ideas for experiments. That would be awesome, and in fact, entirely possible. And, especially those of you in the first year are probably especially interested and kind of hungry for that kind of thing. So what is cognitive neuroscience anyway? I mean, I think that even that is up for grabs. So for instance, have any of you ever seen these big books edited by Michael Gazzaniga called The Cognitive Neurosciences? There's like a new edition of it comes out every couple of years. I think it's in like its fourth edition now. Okay, so if you flick through that, about 2/3 of the articles in it are actually by people who work with rats and monkeys. Which is entirely legitimate because animals have lots of cognition. There's people who work with Jessica Cantlin or Ben Hayden or our chair Greg Daniels know. And but if you go to the Cognitive Neuroscience Society meeting, which many of you have either gone to or will be going to at some point, then it's basically their cognitive neuroscience sort of means like human neuroimaging. And if you look in Journal of Cognitive Neuroscience, it's almost entirely human neuroimaging papers. So given that human beings are definitely not the only people who are the only species that have cognition, that's kind of not quite right. But so I think that thinking about what people do and relating that to what animals do is going to be also valid and useful. And we're very happy that Ben Hayden's going to be giving one of the guest lectures a little bit later in the semester, so that will be a good opportunity to talk about that kind of thing. And there's a kind of caricature of cognitive neuroscience in its sort of brain imaging sense, which is, that's just people who like do bad behavioral tests and give pretty pictures. And it's actually not a very unfair caricature in many cases. So if you feel, and I'm hoping this could be part of the discussion, if you're kind of more from the behavioral, computational end of things and you kind of have this feeling, I don't think the cognitive neuroscience stuff actually really speaks to me very much. I don't really care which bit of the head lights up, which, by the way, you're right not to care which bit of the head lights up, in my opinion. So, what's the point? Well, that's actually a very interesting, valid, and worthwhile point of view, and so it's kind of a challenge to cognitive neuroscience. Well, can we actually present anything that does meet that criterion of being interesting to people like that? We really ought to be able to, and I hope that you'll see over the course of the semester that there is in fact stuff that meets the challenge, but it doesn't come easily necessarily, and there's some aspects which I think are very exciting and promising. And then one thing that I just want to do a little bit near the end today is just show a little bit about searching literature, which is not specifically germane to cognitive neuroscience, but will be kind of relevant to just other stuff you do. And so I taught a class last semester, and at one of the meetings, I was like, okay, now we're going to look at how to search PubMed and Google Scholar, and everyone was like, no, we already know how to do that. That's completely pointless. Stop it. And I'm like, okay, I won. And then people were doing projects, and part of the project involved kind of finding what's the latest work that's been done on this topic, what's interesting out there, what's a good review article. And it kind of came up again and again that people weren't actually really finding the latest stuff, and they weren't really finding review articles, and I thought, okay, well, maybe it would be useful to show how to do that anyway.
Can I just say one thing?
Yes,
so
yes,
I still, on a daily basis, use what you taught us in that class.
Oh, really? Oh.
That. It's incredibly useful.
Oh, good. Okay, well.
Yeah, I had no idea the search terms and everything you could use.
Oh, okay. Well, that makes me feel better.
Right.
Okay, well, hopefully we'll try and get to that.
So,
but anyway.
Syntax is pretty simple.
Good. Good, good. Okay. And by the way, I said, oh, well, we're videotaping the bit where I'm yabbering, but we'll turn it off for the discussion. But really, the whole thing is discussion. So, anyone can say anything at any point, and in fact, is extremely welcome to. This is one thing that I was saying in the meeting last week. I really want to encourage. Well, first of all, I want to encourage you to just kind of turn off that internal veto switch, okay? Because as this is especially going out to the first year students, it's the most natural thing in the world to think like, I kind of have some thought in my head, but nah, that's not a good enough thought to inflict upon the ears of my, other people in the room. But just turn off that switch, because very likely someone else will be wanting to ask a similar question, will actually be kind of happy. Second of all, just speaking sort of opens the floodgates, and next thing you know, people are talking about stuff. And the other thing is, I would really like to encourage just like plain, simple questions. I mean, if you have like a complicated question, that's fine too, but don't ever think like, oh, this sounds like a simple or stupid question, because this is an area in which nobody knows the answers to things. That really is just a true statement. Nobody knows the answers. The simple question, simple things. Well, first of all, it's not even clear what's simple. Even that's up for grabs. But simple things are often just lying under people's noses and overlooked. I mean, there's entire stories in the kind of recent history of how people analyze fMRI data that, for instance, could be framed in those terms. So I would argue that if you're going to do anything useful by studying the brain, then one would hope that it would give you something over and above what you could learn from behavior. And what we really want to try and get at is something to do with mechanisms or representations. And okay, now I don't know if this is going to work or not, but I've kind of wanted to do this in the class for a while. So I thought we might try like a little mini demo of what it is to find an internal mechanism inside someone's head. And this requires you to get into groups of 3 people, or 3-ish people. It's pretty easiest to do it on like physical proximity. If it turns out that I think one will have like 4 people, that's fine too.
Cool.
Okay. So some of you will have done something like this before, but if you haven't, the worst case scenarios will be a bit of fun. Anyway. Okay, so you have to actually like it's kind of sit in some kind of vaguely triangular arrangement. Okay.
Vaguely triangular.
Okay. Now I'm going to come and measure the angles. No. Okay. So this is this. Okay. So one person is going to be the finger mover. Okay. And another person is going to be. You're going to rotate these roles, by the way. The other person is going to just track the finger with their eyes. Okay. And well, the finger mover can do this as well, but also the third person. Just look at the eyes of the person who's tracking the finger. Okay. So you're going to have to kind of stare into each other's eyes a little bit. So just kind of smoothly, slowly move your finger. Pen is okay too. Okay, yeah, just kind of side to side, fairly slowly, and just have a look at someone's eyes. Okay, and just kind of see if you can notice like how they're moving their eyes. Okay, now, and you can kind of toggle between these, and all kind of go around this in different orders, so different people do it. Okay, but wait. Okay, now, but the person who is still like this, keeping the same roles as before. Okay, now do the exact same thing, but there's no finger, okay, or pen or whatever. The person just has to pretend that they're tracking like a moving finger with their eyes. Okay. Okay, and now while that person's pretending that they're tracking a smoothly moving finger with their eyes. Okay, just have a look at their eyes as well, and see and then toggle back to the other one, and then see if their eyes look different in the two times. Then they're actually looking at the moving finger, or they're just pretending to look at the moving finger.
Okay, I'll try tracking something invisible. So I'm tracking something invisible now.
Oh. Yeah. You're tracking you're looking at the eyes of someone while they're tracking an imaginary finger.
Ready?
Eye movement is better than. Did you notice any
Okay, and now just like once you've done that through one cycle, each person gets to like be the pretender and the finger, whatever.
Yeah. Okay. Jumpy? Yeah.
Okay. It was more jumpy before.
Yeah.
It was more jumpy.
It's so strong.
Smoother?
I can't do it.
Okay.
Okay.
Like I feel like with a pen.
That's what I was expecting.
Yeah, I think so. I think like with a pen. Well, Let's try it.
I wonder if you could train
Try
yourself.
with you, okay. So no, so he holds the pen.
Okay.
And you and me switch. So you follow the pen, I look here.
It's basically pursuit scenarios who have not seen.
Okay, cool.
Okay.
I'll try it without.
What we need to do.
So is anyone noticing?
Yeah, go ahead.
Is anyone noticing any differences between the two?
It's a lot different, yeah.
What are the differences?
Pursuit is smoother, whereas you get individual saccades when.
Oh
you're
man! Not only have you
driving.
noticed the difference, but you've encased it in a layer of explanatory jargon, which is actually
It's really unnatural.
But yeah, okay.
You want to try it?
Yeah.
You probably study this kind of thing.
I was gonna say I knew that was gonna happen, but it's still fun to see.
Okay.
Yeah.
Okay.
Yeah.
It's still really cool.
You can probably explain it better than I don't.
I've gotten this included.
The smooth pursuit thing is really strange.
I think freezing the water balloon.
Not bad,
Tell you
but
where.
it's still like pretty complicated.
Okay. Cool. Well, we'll chat about that.
Yeah.
Also disorienting. Or like, dipping the bump.
Yeah, right. Ideally, you're in a black room.
It's actually harder to track.
Yeah.
There's all that stuff going on there.
Michael, isn't this what Kevin did a couple years ago with synesthetes? And he found that even when they're tracking their invisible hand, they're like,
Yeah, so we
still-
used
yeah, you were part of it.
used whether they were smooth or not
Yeah.
as a measure.
Was that like in the
as a
dark
way of
room
measuring.
thing?
Yeah,
Yeah.
So it was a way of telling if they could actually see their hand or not, other than just asking them.
Ah, okay.
Right.
Cool.
The rest
Can
of it
you
was
can you
subjective.
tell people about that in just a second? Okay. But-
Yeah.
Okay, guys. So what did people notice? Did people notice any differences between the two conditions? And what did you notice? What were the differences? Or difference, whatever.
Movements seem much smoother when you are following an object than when you are not.
Okay, awesome. Okay, so what, if anything, can we infer from that about what might be going on inside someone's head? I'm not saying what's a complete explanation, but just something basic like there must be some information going from somewhere to something else or something like that. So, yeah.
One thing that we thought was that when you're following a finger, you're just following the finger. You're not having to imagine following a finger and follow it as well. at the same
Okay,
time.
exactly. Okay,
And
excellent.
then Amanda had also thought
I had some thoughts about like possibly you make some predictions about the movement of the finger through space. And so it's not the case that you necessarily need to like move between points. You can just move to the end point of where you expect it to be in a movement.
Okay, that's very interesting as well. Yeah.
To generalize a little bit, you can say that there's some really strong feedback mechanism from your concept of the world or representation of it back to the muscles that control your eye.
Excellent. Awesome. Okay. So basically, there's some information going from your retina. Now, what that information is and what happens to it when it goes from your retina is an open question. Okay. But somehow there's some information that goes from your retina to whatever controls your eyes that's actually being used and which is helping them to move more smoothly. And if you don't have that information there anymore, in other words, you're not actually looking at a real finger, it doesn't move as smoothly, and in fact, I guess it's kind of impossible. Is it impossible to move it as smoothly? But okay, so there's some mechanism in your head that's moving some information from some part of your head to another part of your head. Okay, and you establish that just by looking at people's eyes and waving a finger around. Okay, no MRI machine was involved, no computer was even involved. Okay, so you found something out about an internal mechanism. Okay, and now as it so happens, we happen to have right here. Do you go by Mike or Michael?
Usually Michael.
Michael Melnick, who actually knows a thing or two about this stuff. So tell us a couple of interesting facts about smooth pursuit.
So it is entirely the case that you really can't smoothly pursue with no object to track. A bunch of this has been linked to the frontal eye field, the FEF, in kind of linking the motor behavior and visual information. But we used this as a measure of actually if you could see your hand or not. There was an experiment we did in our lab where we found that if you completely blindfolded someone and asked them to wave their hand in front of their face, typically still people report a visual percept. And it depends on the person how intense that visual percept is. Sometimes it's just like kind of a block or a bar that moves in front of their face, and sometimes it has individual fingers depending upon the person. Synesthetes have really strong percepts. But one of the versions of that experiment we did, we tracked for smooth pursuit eye movements because It was a way for us to empirically measure if people were seeing their hand or not, rather than just asking them subjectively if they saw it. And so that was, yeah, that's one way you could use it, was to kind of track back to see if someone actually sees something.
That's very interesting. Thank you. By the way, Michael works with Due Tadin, who is also going to be giving a guest lecture in the class. Very happy to say. Now that last bit was especially. Oh, by the way, if you want to read more about that, go to Google News, because that stuff got written up in the news quite a lot, right?
Yeah, that was Kevin's study. That was
Yeah.
a former
Yeah.
grad student.
Yeah. So, okay. Now that last bit was especially interesting. That you were able to. Okay, so you wanted to make some inference about. Were people really doing something like real eye tracking, or were they just, imagining, pretending, or whatever? Okay, so there's some hidden process going on inside their head, and you wanted to take some measurement from outside their head and make some inference about, what's going on inside their head. And so you said, okay, well, if there really is something really akin to tracking genuine, to real smooth pursuit going on in their head, then they ought to be moving their eyes smoothly, and in fact it's kind of impossible or very difficult or impossible or something to move your eyes smoothly without that real input, although there's not actually input in that case, so it's what the internal signal is an interesting question. But so that's okay, so now you're making, so not only are you kind of figuring out that there's some kind of mechanism, some information is going from one place to another place, is being used for some task, but you can even kind of leverage, I guess you say in this country, that to actually say, aha, if we observe that such a, that this kind of external sign of that process is happening, namely that people really are smoothly moving their eyes, then that means that there really is something internal going on even when one might not expect it, such that, it kind of looks like people are smoothly tracking their finger even when there's no physical way they can see their finger because, they're completely blindfolded. So there's all kinds of interesting, so that, that's cognitive science, right? That's kind of trying to figure out what's going on inside someone's head, how information is being processed, what's being represented, how it's being used. And none of that used any, no electrodes were stuck into the heads of any people, animal, rats or monkeys. No scanner was used, no computer was even used. Used an eye tracker, I guess.
Yeah, that was a fancier version of the
Yeah.
experiment that actually also used full field noise.
Okay.
It was effectively eye tracking.
Okay, cool. So, so from outside the head we can figure on something that's going on inside the head. So then you say, oh, well, if you want to step that up a notch, just actually let's look inside the head too, and then we're really going to be in great shape, right? Well, maybe if you do it right, but also not if you do it wrong. And so that's one of the reasons why I put this this reading about why you should be skeptical of brain scans. And this guy, Michael Sherman, I think he's the head of the Skeptics Society or something, so he's kind of a professional skeptic. But everything he says is extremely reasonable, and here's some of the reasons why you should be skeptical. Oh, this reminds me actually, did you guys all bring little printout thingies? It's not too late to run and print something off if you didn't, but that's kind of meant to be part of the evaluation that we pick up your printout. It's basically just to kind of, it's got two functions. One is to help make sure that people actually do the readings, and the other is that if you kind of draw a blank when people are discussing, you can just look at your piece of paper and say, well, I wrote this down, So anyway, so here's some reasons. Here's some reasons why you should be skeptical. How many of you have actually ever been in a scanner or subject for okay, not just even for an fMRI study, but just for, had an MRI scan or any kind of, okay. So if you've ever been, you're in this tube, it's very strange and weird. In the article he says how he got claustrophobic really quickly, which is actually not an uncommon thing, even if you've never been claustrophobic in your life. So let's talk a little bit about some of these, just some kind of problems and difficulties with brain imaging, and this is going to sound negative, but it's ultimately, I hope, not negative, because if you're going to do interesting stuff, it's going to be by being fully aware of the pitfalls to not doing interesting stuff. And luckily, I think that there's plenty of scope to do interesting stuff beyond that. Okay, so here's a kind of caricature, and it's really not a very unfair caricature in many cases of a bad brain imaging study. You say, well, let's stick people in a scanner, get them to do some task X, preferably some really, really complicated task, like doing New York Times crossword puzzle or something. See which part of the brain lights up the most and say, this is the part responsible for X. And in fact, that's often how these kind of things get reported in the popular press. If you go to Google, this is like this autocomplete that I got from typing in part of the brain responsible for it starts. Actually, this is kind of a fun game in general, like what Google Auto does. You've probably seen this, right? Like, there's even the whole website's devoted to this. It's basically like it's a way of sort of measuring the world's collective prejudices, which it can be a bit scary. Okay.
It actually trains to you too. So sometimes it's best to try looking in incognito.
Oh, really?
It trains to what you previously searched for.
Oh, okay. I don't know if I was signed in or signed out of Google when this happened. Okay. Well, maybe this just tells you about what I would like to know the answer to. I don't know. But okay. So some of these are kind of legitimate-ish, like responsible for speech. Ben Hayden's going to talk all about decision making, and you'll see that there's many parts of the brain. In fact, that's true for all of these things, and that's going to be kind of a theme. Part of the brain responsible for love, well, that's a very high-level complex thing. It's not really the case. It's easy to think that there might be a part of the brain that does X, but that's probably not a very good way of thinking about it. Or is it? Well, we're going to see some discussion about that too. Okay, so he talks about, well, there's this idea that things are modular, and it's very easy to kind of think that that's the case. And to say something's modular, well, even that's something that people don't necessarily agree on, but it's something that's kind of self-contained, that you can sort of join them together and they do their own thing. So it's reasonable to say, for instance, that organs in the body, like that. And as people use this metaphor, the brain, are there things like cognitive organs in the brain? Is there a language organ in the brain? So your kidneys are obviously connected to your heart and your liver via many pathways, but they're also kind of, you can transplant these things, right? It's not easy, but that kind of shows that they're modular, right? And then there's other things that are more distributed, like blood circulation or immune system is a really nice example. So there's a theory out there which was initially popularized by Jerry Fodor, who's actually a philosopher of mind, that the brain is modular, and this is actually still an influential and hotly debated topic, as we'll see in a minute. And then but it does also come down to questions of interpretation as well, because you could have things that are sort of functional modules in the sense that they have a set of things they do, but they're all over the place. So in that case, you'd say, yeah, fine, the blood circulatory system is a module because you can actually, in theory, separate it from the rest of the body, and it certainly has a function or a set of functions. So anyway, as is probably apparent, I personally don't really believe that much in modularity, but that is of no consequence. What matters is, what you guys think about it, and certainly there's other people who think differently, including people who are, very well known and eminent, and we'll see some in a minute. So here's a kind of metaphor for how I think about it, which you do not in any way have to agree with. And Frank, every single one of these slides you've seen before. Just you're already thinking that.
I have bad memory.
Oh, yeah, I don't think that's true actually. Okay, so suppose I said, what's the Google autocomplete? What's the part of the brain responsible for love? Well, you could, and people actually have done these kind of studies. Put someone in a scanner, show them pictures of their romantic partner, show them pictures of normal people, see what the difference is, apply some kind of threshold, and you will get some areas, okay? Probably emotional related areas, and you say, okay, these are the parts of the brain that where love is. Okay? So to my mind, this is my own personal metaphor that I kind of like, but you can see what you think of it. If you say, where in the United States is the economy? Okay? Well, the economy is kind of a bit like the immune system; it's sort of everywhere. Okay, but it's actually very reasonable to say, suppose you say, okay, we're going to take the United States and apply a statistical threshold and find the sort of, most statistically significant hotspots of economic activity. You'd say, aha, we've found this place in Lower Manhattan called Wall Street, and it turns out that that's where the economy is. And that's actually not a ridiculous claim. So for instance, you could say, well, let's test that. Let's go and lesion Wall Street and see what happens. Okay, and that's kind of what happened in 2008, right? Wall Street got lesioned, and it had big effects on the entire economy. People in, Dakota were losing their jobs because of what happened in Wall Street. Okay? So there's a sense in which you can have, sort of hotspots of concentration, but you would really be missing an important part of the picture if you said the economy is in Wall Street. Okay, although you wouldn't be saying something completely false either. Once I was saying this and talking to someone said, well, although it's the case that there's places that are not in Wall Street that have the economy, it is the case that everything in Wall Street is, the Wall Street itself is 100% specialized for the economy. And that's kind of true, but it's not completely true. I mean, there's hot dog stands in Wall Street, right? There's, there's a subway station, okay? So there's, even the most kind of unifunctional parts of the brain actually have multiple functions. And in fact, this is a theme that's come up over and over again. People think, some brain area does X. Like, if you say to someone, what does the amygdala do? People will say fear. Okay, it turns out the amygdala is involved in lots more stuff than fear. Now, a lot of what the amygdala does is fear, but it goes significantly beyond that. So one way of looking at that is you say, okay, don't just look at little hotspots, look more broadly. So you can say, well, let's look at broad patterns of neural activation distributed across multiple areas, and that's what a lot of people, including me, do, and that's an approach which is quite popular these days. So that's one way of getting away from this sort of perhaps oversimplified modular view. So some other points that Sherma says, MRI is a very indirect measure of fMRI is a very indirect measure of brain activation. So I just want to very quickly spend a couple of minutes just showing, kind of going through why that is the case. And many of you will know this already, but I won't spend too much time on it. So MRI stands for magnetic resonance imaging. If you want to learn about the physics of it, there are classes on that, but this isn't one of those. But essentially, it takes a 3D picture of the inside of the body. It's non-invasive. It doesn't actually involve slicing people's heads open. You've got two types of scans, essentially. There's actually more than two, but the two main ones, the purely anatomical one, which is what this is, it just shows the structure, or you can stick this magical seeming F in front, which actually, I don't like the name function because it doesn't directly show you the function. It shows things changing over time. Okay. Now, if you want to get to function, seeing things changing over time is certainly a step along the road. But it doesn't sound as good to call it changing over time magnetic resonance imaging. Okay. So it shows brain activity indirectly. And what does indirectly mean? So you've got like this little kind of cartoon strip of brain images. Usually it's actually every 2 seconds. I should update the slide. And you've got a movie. But what we're actually measuring, and again, it says this in the article, you probably already know this, is you're not measuring brain activation directly, you're measuring indirect changes that are actually, why can you measure anything about brain activation at all with a big magnet? Because luckily for us, it just turns out that little magnetic changes actually happen when your brain is active. The reason, why is it that little tiny magnetic changes happen? Because when neurons get active, extra blood gets pumped there with extra oxygen in it. Like if you go to the gym or something and you sort of like lifting up a weight and you look and you see there's like, veins in your arm or whatever. Yeah, Zach.
Might be a slightly stupid question, but how does the body know where to send the blood? Okay,
I'm so happy that you prefigured that with saying that, because this is a perfect illustration of actually a really good question. And everything that seems like a too simple question is in fact almost certainly going to be a really good question. Okay, so anytime any of you find yourself saying, this might be a stupid question, try to mentally flip that to, this is probably actually a really deep question getting at something important, because that actually is. Okay, because that is, the question is, how does the brain know where to give the extra blood? That is a very, very actively researched topic to which people do not completely know the answer at all. And there's probably entire conferences, certainly entire workshops and entire subparts of journals devoted purely to that question. There's a very, very interesting question. It probably involves, I won't even go into it, but there's a lot of stuff in that. There's like these kind of helper cells in the brain called glial cells that kind of carry signals around between the neurons and sort of connect the neurons to the blood capillaries, and they probably carry some signals. So anyway, so the area gets extra oxygen. Yeah.
One more question.
Yeah.
So, and people are like 100% positive that when you see this increased blood flow, it's always like directly related to increased neuron activation?
Also, an absolutely excellent question. And so, no, people are not 100% positive about that. And in fact, for a long, well, this is a whole story in itself. These are really great questions, and they're both kind of simple and get directly to the point. So, people were doing fMRI for a long time, and they were saying, well, here's a kind of reasonably plausible story saying that these things, that this extra oxygen is probably tracking extra neural activation. And if you do very, very sensible and reasonable things, the kind of answers that you expect come out. So for instance, if you show somebody a flashing checkerboard and really make their visual cortex super active, you observe extra oxygen in the visual cortex. If you don't show anyone anything visual and you play sounds, you see stuff in their auditory cortex. Okay, so if it isn't to do with neural stuff, then the brain's really trying to trick us. But still, that's still an indirect argument. And it wasn't actually until people have been doing fMRI for about 10 years that a guy called Nikos Logothetis this wasn't really kind of conceptually a difficult thing to do. It was a very technically difficult thing to do, to record single neurons' actual neural activation from monkeys at the very same time while they're in a scanner. So he built like a whole special scanner to do this. It's very, very technically difficult setup and cost lots of money and was scientifically challenging too. And said yes, these things actually do go together. Although the correlation is not perfect, and there can totally be times where you see stuff that looks like there's a lot of blood-related things happening, and it's not really to do with the brain. So a classic example is if you have a big vein, and your head is full of like big blood vessels, both arterial for bringing the oxygen in and veins for taking the blood away again. And you can pick up huge signals from them that you're really picking up the plumbing much more than you're picking up the neural activation. I mean, there's ways of trying to guard against that. Yeah.
So I guess that goes with my question, which is exactly how does the brain get blood in different parts of the brain?
So so this is really a huge question. So there's a very, very rich and fine-grained blood circulatory system in your brain, and it has all kinds of controls in it that people don't fully understand.
Do we also consider that to be a measure, like the pattern for which blood flows through the brain to the different areas?
Yes, that's right. So in fact, there's different types of scans that you can do, and standard fMRI measures the oxygenation in the blood, but there's other types of scans you can do that specifically measure blood flow. People sometimes talk about fMRI as if it measures blood flow. It measures something that's extremely similar to blood flow and very highly correlated with blood flow, which is blood oxygenation. But there's ways you can measure flow directly, in fact, the very, very first scans that people did, so currently the way you don't have to get injected with anything when you do an fMRI scan, luckily, because your blood oxygen, the hemoglobin actually happens to serve as the carrier of the signal. So lucky us, we have MRI signal already flying around us. But before people figured that out, they actually injected people with what's called a contrast agent, which is like a kind of extra MRI signal giving stuff, called gadolinium usually. And that actually does, you literally kind of see that flowing around in the brain. So that's what people use first of all. Yeah.
So another question is, do you have to do this on an individual basis determine the differences in oxygenation? So like, for example, if you're dealing with someone who's anemic or someone who regularly lives at high altitudes and you scan them in a lower altitude.
Right, that's a very, very good question. People have probably done an altitude study, and I don't know. That would be something that would be fun. That people also look at that as well. So you can even kind of try and calibrate the effect. For instance, if you get people to breathe in air that has a little bit more carbon dioxide in it than usual, that actually affects this in a kind of predictable and measurable way. So you can kind of, slightly modulate the air people are breathing and kind of get this plot of, effective oxygenation. If you give people caffeine, that actually slightly, if I remember rightly, constricts their blood vessels, and so that has an effect too. So it can vary. If older people tend to have, unfortunately, worse circulation systems than younger people. There's a lot of
pre-screening questions you ask people before you scan them, like have you had coffee in the last hour?
Well, it turns out because MRI is a big magnet, the only really, really, really important thing is that people don't carry metal in there. So most of the pre-screening, if you ever do one of these scans, they ask you in 100 different ways, do you have any metal in your body? You get rid of all your, rings or earrings or whatever, or piercings, but, it'll also say, have you had an operation? Have you ever worked with, like, welding equipment, stuff like this, okay? So as long as you don't have metal, particularly iron-containing metal, then you're not going to really hurt anybody. And then it's just a question of how good will your signal be, and that depends on lots of things. It mostly depends on how well people keep their heads still. But all these other factors that you mentioned are important too.
I think there are a lot of places, like I know we definitely ask people, at least we ask parents, like if their kids have been diagnosed with like ADHD or anything that could suggest that they're on a medication that could affect blood flow or neurotransmitters that secrete in the brain. So I think that's pretty typical.
Yeah, and that's a very good point.
Those kinds of questions.
Right.
Yeah, and that's true of our stroke patients too. We screen them for all sorts of antidepressants and all those things, and then we also actually run a separate set of scans to try and de-warp their images, because we know that because they have extra airspace, it's going to warp all of our blood flow images too. So, yeah.
That's a very interesting point. And I mean, for these kind of drugs, there's kind of two things. So the drug might be having a direct effect on the circulation of blood oxygenation, and I don't know if they do or not. They might do. Probably some drugs do. But also, typically, you just kind of want your set of subjects to be, I mean, depending on what your study is, but you often want them to be kind of as normal as possible. So you don't want to say, we found some really interesting fact about how the human brain in general works, and you don't want someone to say, no, I think actually you found an interesting fact about how a particular subset of people who happen to be taking some medication work because you didn't, screen your subjects properly. Now, it might be that, in fact, you're really interested in, say, studying kids with ADHD, in which case you would apply these screens on your control group, right? But so trying to figure out like who you're actually, and this is true not just for neuroimaging. This is true for any kind of study. You want to have a pretty good idea of whether the people you're studying, have kind of important characteristics that you ought to know about but don't know about, right? Okay, so we're measuring blood, and as it turns out that actually more oxygen gets sent to an area than is in a certain sense required. For a long time this was a mystery, although apparently people just think now it is that if you really want oxygen to diffuse quickly through, if your body really wants oxygen to diffuse quickly through the capillary walls, then the best way to get it quickly delivered is just to have loads of oxygen in there. So it's not maybe that mysterious that extra oxygen goes there. But anyway, extra oxygen does go there, and so that has the effect of making the MRI signal higher, it turns out. Okay, so that's actually what gets picked up, and it's called BOLD imaging, blood oxygenation level dependent imaging. So there's a sense in which it's an indirect measure. But there's another indirect measure, because any of you who work, do any of you do actually neurophysiology? Or planning on doing neurophysiology? Okay. Yeah, you're all working with Ben, right? You're just putting your hands up. Okay, so you're going to be, sticking electrodes in actual, if not within individual neurons, and very close to individual neurons, and trying to pick up spikes from individual neurons. And so even a few millimeters of brain tissues, has millions, tens of millions, hundreds of millions of neurons in it. Okay, so it's really, and moreover, the information is getting processed by very intricately connected populations, those neurons, and we're certainly not picking up on those intricate connections either. Okay, so it's again indirect. Okay, I don't want to spend too long on this because I want people to kind of have discussion more. But so the simplest possible design is you say, I'm looking at how things change over time, so I'm going to put someone in a scanner and make them do something that changes over time. So, for instance, you say, okay, when the screen comes on, I want you to, wiggle your finger, and when it says rest, I just want you to rest, or whatever it is that you're getting them to do. And then you can arrange that in the simplest way in some sort of block of 30 seconds doing tasks, 30 seconds resting. You don't have to do it that way, but that's a simple way of doing it. And then you say, okay, we're going to look at the signal from different voxels, and if some of them sort of go up and down in a way that seems fairly correlated with that, then you'd say, yes, this is an active voxel, and if you have ones that, they might be doing something, but whatever it is, it's not really correlated with your task, you'd say, well, that's an inactive voxel. And so Shermer also makes this point, well, when you see these brightly colored blobs, that's not actually you don't actually get these images out of the scanner. This is a kind of compound picture that represents two different things at the same time. The gray stuff is something that the kind of high-res looking underlay that you actually do get from the scanner. But much lower resolution is the functional data, and when you see some kind of brightly colored blobby stuff, that's actually been statistically processed, so the brighter it is, the better this correlation is, and then you stick some kind of threshold on it, and you say, okay, I'm not going to show you the dim stuff. So it looks like there's only things happening here, but in fact there's stuff happening everywhere, it's just that it doesn't reach some significance criterion everywhere else. Okay. So there's another sense in which things are indirect. Okay, this all sounds kind of depressing so far, but it will get less depressing in a minute. Okay, but before that, a little bit more difficulties. So even if you get some activation, then the really interesting, well, difficult but interesting problem arises of trying to figure out what it means. Now, the activation doesn't come with some little label attached to it saying, "Hello, I'm activation corresponding to, your favorite cognitive process." It's just some activation. Okay. And it's actually quite difficult to figure out what cognitive process it might involve, because brain areas do lots of things. So, for instance, and this is just a subset of the things that parietal cortex does, any one of these processes could be activating parietal cortex. So if you observe parietal cortex being active, then it's hard to know which one of these it actually was. Now, it might be that you're doing an experiment on attention and you really love attention and you care about attention. So when you see parietal cortex active, you're going to think, aha, someone's, must be paying attention, okay? But, the brain doesn't really care what you happen to be interested in. Any of these things might have done it. So people often call this reverse inference and say, well, this is kind of a dodgy thing to do because there's many things that may have caused a given activation. So that's a little bit abstract and specific to brain imaging, but this actually happens in real life as well. This is a famous example. I'll skip that, but it's okay. So here's an example of reverse inference in real life. So if you have bubonic plague, I could cross this out and say Ebola virus, okay, to be more current, okay. Then you'll experience flu-like symptoms. That's I don't actually know if that's a true statement, but it's probably a true statement, okay, right? And then you say, oh, I have flu-like symptoms. Okay. Oh no, and I don't know, but I sometimes find myself thinking like this. But you probably on occasion of if you ever Google for some disease, never Google for a disease, right? Because you'll think that you have
the worst thing in the world.
Okay, so why? Because you see, you say, okay, if there's such and such disease, you'll experience some symptoms. You observe you have these symptoms, and you think, oh no, I must have that disease. Well, why is that an invalid inference? Because loads of things could cause those same symptoms, especially something like flu-like symptoms. Everything causes flu-like symptoms. Okay? And that's the exact same reason why reverse inference is something to be careful of in brain imaging, or in fact in any type of making an inference about some hidden process. Okay? So before we were talking about from the fact that people are moving their eyes smoothly, we know that something's happening inside their head. Whatever that something is, it's something that involves information going from the retina. Well, it probably involves information going from the retina to the eye control circuit. Well, due to the work that Mike was telling me about, it seems that can happen even if there isn't information going from the retina. So, it can be difficult. There can always be kind of extra hidden factors that you haven't thought of. I mean, that's true just in science in general. But it's not even just that there's different cognitive processes that activate the same area. Even if you have the same general overall process, like speech perception, okay, there's loads of different neural representations, all of that same processing type, that are all going on within that area. So, for example, and I've kind of done this kind of study, for instance, if I'm having people listen to sounds like ba and da, okay, it makes perfect sense, and in fact is the case, that your brain's going to use the same kind of machinery for processing the sound bars it uses for processing the sound da. I mean, it would be just incredibly wasteful to have like separate resources for that. There's not a bar area over here and a da area over here, so they're all in the same area, but they're slightly different populations of neurons wired up slightly differently. Something must be different because they're sensitive to different sounds. So if you stick someone in a scan and you play them bar, their speech-sensitive brain areas, auditory cortex, Wernicke's area, etc., will light up. If you play them a different sound, but still a speech sound, the very same areas will light up. So if you say, aha, I observed the exact same degree of activation when someone hears bar and when someone hears da, well, if I subtract them, I can hereby conclude that there's nothing left, so I guess this brain area is not sensitive to the difference between bar and da. Well, that doesn't follow at all. It just means that the way in which it is sensitive is not something that you're picking up from just looking at kind of how much the things light up. So when different representations are kind of present in the same brain area, which is, by the way, always the case, okay, brain areas all do lots of things and all have lots of different representations inside them, then you have to worry about, well, how are you going to try and pull them apart? Now, there are, in fact, ways of trying to pull them apart, which we'll talk about later, but it's a challenge, okay? So, in fact, if you go to, well, actually, this is probably not quite as true anymore as it used to be. In fact, this is not as true anymore as it used to. But back in the day, if you did a PubMed search or something like that for representations in fMRI, and this is still what you'll get in the most part, you will get localized and segregated representations. Okay? So sometimes the brain is really kind and obliging and makes things nice and easy for you. This doesn't happen very often, but it does happen sometimes. And it actually has representations of different things in different places. Okay? So for instance, you've probably all seen this sort of little homunculus kind of map of like how your body and muscles are represented along this sort of somatosensory motor strips along here. Okay, so it really is the case that hands are sort of up here and lips are sort of down here. Okay, so you really actually can look at which part of that motor cortex is activated and say, okay, that must have been someone's hand doing something or their lips doing something. Okay, and even in more high-level type phenomena, and you're going to hear more about this. Those areas of, say, ventral temporal cortex. This is actually a view of like the bottom of the brain. Okay, you're kind of looking up underneath someone's head here, like as if you're looking up through their neck or something. So there really are some areas which tend to be a little bit more special. Tend to be more specialized for exactly what that means is up for debate, but tend to respond more to, say, houses and dwellings and landscapes and stuff like that, and other areas that tend to respond more to things like faces, and they're actually in different places, such that you can look at someone's brain, see activation in one of these areas or the other, and actually figure out that someone's more likely to be looking at a house and more likely to be looking at a face, and you'll be right as well. Okay, but usually life is not that easy, and in fact, even in these cases, life is not that easy. We'll come to that. But much more generally, yes. Oh, okay. We'll take it in turns. Yeah.
Never mind. So what you were saying about looking at a brain scan and being able to use that to predict whether what they're looking at or something.
Yep.
So is it that when you can do something like that, it's a very strong indicator that it is segregated?
Not necessarily, because it turns out you can. That's a very interesting question. It turns out you can do that even for overlapping representations. And in fact, I'm actually about to show you, well, no, I'm not going to go into the details, but I'm going to show. So when you have, okay, so when things are local, but it's a very good question. It just means that things are distinguishable, okay? But for things to be distinguishable, they don't have to be in separate places, right? Like, I can tell that, your face is different from Alyssa's face, and one reason is because you're, in different parts of the room, right? But if there was like a TV screen that was like flipping between your two faces, I'd still be able to tell, right? Now, the way it'd be more difficult, you might, if you wanted to have some computer vision algorithm do it, it would have a tougher time when they're overlapping than when they're in different places, but you'd still be able to do it. And so that's not actually that dissimilar from how it would get done in an fMRI analysis as well. So localized and segregated representations are nice and easy, maybe misleadingly so, but distributed and overlapping representations are very common, and a famous and important paper, which we'll probably talk about later in the course, is this one, for instance, that came out in Science 2001 that is specifically about distributed and overlapping representations, and it could tell them apart even though they were sitting on top of each other actually by looking at the pattern differences between them. Okay. So if you do have all these representations, so suppose I've got all these different speech sounds and they're all, happening in the same kind of speechy part of the brain, in the Wernicke's area, they're basically all sitting on top of each other. You've got this kind of representational pile-up. So it's a non-trivial problem to pull them apart, but it's actually something that can be done with some success. And one way in which you can do that is by looking at, not just looking at the total amount of activation, but by looking at the pattern differences. So suppose I have, stimulus A that produces, this is a kind of cartoon of some voxels. If I have stimulus A that, makes this kind of pattern across those voxels where the brighter stuff is more active, well, I can just jiggle around those squares so the total amount of brightness is exactly the same. The total amount of local activation is the same, but I've just changed the pattern a bit. So now I've got a different stimulus. Well, if you had some kind of algorithm that said, well, let's look at the pattern difference between these things, you can actually tell what the difference is, right? Even though if all you did was just add up the total amount of kind of bright yellowness, you'd get exactly the same answer. Okay? So that's an approach that people do quite a lot. So, okay, so let's just kind of quickly think about the other paper, which actually kind of has some interesting conflicts with, I don't know if you guys noticed this when you're reading, but there's some kind of interesting conflicts between these two papers, which goes to show that this is an area full of debate and open questions, like right from the get-go. Like I didn't have to work very hard to find papers that had conflict with each other. Okay? So this is Mather, Cacioppo, and Kanwisher. And Nancy Kanwisher is actually very well-known and eminent cognitive neuroscientist who's probably the primary, but far from the only, but probably the most prominent proponent of this kind of more modular view, that there are actually areas that do like particular things. And she in fact has sort of discovered some of these modules, so where it's an open question. Not everyone accepts that they are modules, but there's certainly something going on in there. And this is from another reason why I picked this paper is it's actually very, if you decide that you're interested in these questions, this is a really nice special issue. Like this is, I think, like the introductory article in it, but there's a whole bunch of articles in there addressing this very good and important question, 20 years of fMRI, what has it done for understanding cognition? And there's some people who basically, perfectly reasonable people, who basically say, yeah, it hasn't done anything for understanding cognition. Okay? So it really is a live question saying, well, just how can fMRI inform cognitive theories? Okay? So, I was saying, oh, this sounds so depressing. There's all these reasons why it's really hard to interpret brain activation. It's really hard to figure out what's going on. It's hard to, make these studies actually tell us anything interesting about cognition or about representation or about information processing. Well, there's people who are taking up that challenge, a lot of people, and saying, well, no, hey, there are some ways. So they suggest a few ways, and this is far from the only ways, and these are not even necessarily ways that everyone, including me, agrees with, but I think they're interesting to think about. So they highlight 4 different types of questions. So one question is, which functions can be localized to specific brain regions? My personal view and view of a lot of other people, it doesn't actually matter very much what my personal view is, but a lot of people think, well, nothing can really be localized. There might be Wall Street type hotspots. But Kanwisher and these other authors say, well, no, actually, maybe you can localize some. Can you find a marker of a mental process X? Okay, well, now that's an interesting question. So, if you're seeing someone's eyes and they're smoothly moving rather than kind of moving in a jerky manner, there's something going on in their head that's making that happen smoothly. Now, it's not necessarily clear what that is, but there's some, in this case, kind of, visual motor process that's happening in their heads, and you've got a marker of it, okay? An externally observable marker because you can see that their eyes are moving smoothly. How distinct are different representations? Do they very similar question, do tasks engage common or distinct mechanisms? So let's just quickly look at a couple of examples of that. Then I'll spend like 2 minutes showing you some search tips, then we can have a bit of a break for coffee, and then we can chat a bit. Okay, so which functions can be localized to specific brain regions? Well, here's a quote from them. They say, the brain does in fact contain some specialized mechanisms for particular mental processes, e.g. the fusiform face area and some other areas. Okay, this is a controversial claim. Lots of people accept it, lots of people don't accept it. There's entire reams of debate about these kind of questions. Okay? A lot of people are kind of coming to the view that these are areas that, yes, they have greater sensitivity for certain things, but they're not really like kind of pure specialized modules. And that can actually be shown empirically, depending on your point of view. Okay. Can markers of a process be found? So this is, one of the really interesting questions. You could argue that, a lot of cognitive neuroscience basically boils down to trying to do this, right? Trying to look at someone's brain activation, trying to figure out, well, what does this actually mean? They give a couple of examples where, and they cite papers that suggest them, which I actually think are valid, where because you can see something happening to the brain activation, you really can actually infer, well, there is some process happening. So for instance, if the stimuli that are being actively ignored, you can actually see that their activation gets suppressed, which suggests that the representation is getting suppressed. You can look at how attention directed to one aspect of an object sort of increases the activation of other aspects of that same object, so it's kind of like it spreads across the whole object in a certain way. There's quite a lot of nice examples like that, and they give some of them. How distinct different representations. This is a whole discussion in itself, but this kind of relates to your question of, can you actually tell two different things apart? Like, if you're trying to figure out, is someone looking at a face or a house, that's kind of easy because those stimuli happen to give spatially distinct representation. But in fact, you can even get ones that are spatially on top of each other. You can still tell the difference to some degree. Now here's an interesting one. Can you figure out if two different tasks engage common or distinct processing mechanisms? And they have this nice line here. They say, if conducted properly, you have to be careful here, but if conducted properly, experiments showing overlapping brain activation with controls, blah, 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 can provide evidence for common mechanisms. And they give a very interesting example that, people just kind of observed. I don't know enough about memory research to know if this is historically the way that things actually really happen, but it probably is. People hadn't really thought that much about how remembering the past and imagining the future, they're probably related, I guess, but people just hadn't really thought about that much. And then people noticed that actually they seem to activate exactly the same kind of hippocampal brain areas. And then people were like, well, hey, maybe this is really kind of the same process. Maybe the whole point of memory is to actually help you imagine the future. There's a guy called Dan Schachter who is very prominent in that area. Quite a lot of people work on this. So they went and tested that, and in fact it turns out that, if you get people with memory deficits and you ask them to imagine the future, they're much worse at it, which is kind of an interesting, surprising result. So here's an argument from, so I like that as an example, here's an argument from overlapping activation to saying, yeah, there's something actually connected in the mechanisms. But now here's a slightly, but maybe not completely tongue-in-cheek counterexample. Okay, so when people talk, they use their mouth. When people eat, they use their mouth. Therefore, talking and eating are the same process. Right? Okay, well, obviously that's not true. But there is a relation between talking and eating, right? I mean, they do both use your mouth, and there are some commonalities between them. But it just is an illustration that, you've got to be a little bit careful. And the authors of this are actually fully aware of this kind of idea. They say you've got to be careful, but what does being careful actually mean? When is this a valid inference and when isn't it? So these are, I'm really just trying to kind of raise questions to think about here, but the punchline is, there's lots of open questions here. Even something that looks like a simple question is actually probably getting at something deep and interesting. And there's lots of controversies and kind of fun and open questions to think about. This is probably true in almost all areas of science, but I guess there's some areas of science where people kind of have a lot of it figured out and they're just trying to kind of paint in the details now. Maybe, I don't know, maybe some parts of particle physics are like that. I don't know if that probably isn't even true, but cognitive neuroscience is not like that at all. Okay, so things that you can't walk 5 yards without hitting an open question, and from the point of view of like a new student looking for research, that's hopefully kind of exciting. Also, may be a little bit overwhelming because you're like, oh my god, there's like so many things, I don't know what to do. Okay, so that gets to this point about. Let me just quickly show you a couple of tips for like how to find stuff, and then we'll take a little break, and I can we can give people some coffee. Okay, so I just want to show you a couple of things. Okay, so let me pull up, hang on, let me change the mirroring on here. Okay. Okay, so my suggestion, now you'll have noticed from the readings that a lot of the readings are from a very nice journal called Trends in Cognitive Sciences. We are very lucky. There exist really nice journals that specialize in writing short, comprehensive review pieces by experts. Well, now I'm not saying you can just read a 6-page article and you know everything about the field, but you'll have at least a kind of pretty good roadmap and you'll know where to look next. And that's a nice thing to have, especially when you're like, oh my word, there's like, you know, I mean, if I type something in, you know, say you're interested in, I don't know, attention and, you know, cortex or something, okay? I don't know how many hits this is going to get, but it's going to be a lot, okay? Okay, I've got 15,000 hits, okay? So if you're interested in attention cortex, just read 15,000 papers and, you know, we'll talk about it next week, okay? So that's not going to help you very much, okay? But, you know, there's review papers. Now, it turns out that PubMed is especially helpful here, okay? Because it actually tags review papers with the word review. Google Scholar doesn't do that, by the way. Google Scholar will pull up papers that just contain the word review, and you can have papers that contain the word review that are not review papers, okay? And you can have review papers that don't contain the word review, okay? But in PubMed, you will actually, well, there's even a little, you can even click it here, or you can type it right in the search box, okay? And now I'm only going to get review papers, okay? Now I still have 2,200, okay? But this is already helpful. Now, if you happen to already know that you particularly like Trends in Cognitive Sciences, that's not the only good review article, by the way. There's a journal, there's other ones. Current Opinion Neurobiology is very good too. These journals have horrible names. They're wonderful journals, but they're called Trends in blah or Current Opinion in blah, as if, you know, like, you know, fad chasing in blah, you know, I mean, but they're great journals. I wish they were called something else, but they're not, but what can you do? Okay, okay, so how are we going to narrow this down a little bit? Here's, so many of you have probably done this before, and now at this point, you know, in my last class, everyone was like, yeah, we know that. And but it turns out that you can also do all this kind of Boolean searching, meaning you put in ands and ors and stuff, and that can actually help to narrow things a lot. Okay, so suppose you say, okay, I care about attention and you know, objects. Okay, but I don't know if people are going to talk about objects or object or something, so I can put object star. Okay, and I don't know if people are going to talk about cortex or brain. You know, what if they're talking about the brain but they don't use the word cortex, or, you know, maybe I'm interested in you know, thalamus or something. Okay, right? So you can do this kind of thing, right? And now I still have a lot, so I can put in a few more ands. So say I'm interested in you know, visual stuff, right? Visual attention. Well, I don't know if they're going to talk about vision or visual, so I could do, right? And you can do the brackets and or parentheses, I guess, and I think you have to put these Boolean bits in capitals, but you can start to narrow things down a bit. Okay, but PubMed, the single most useful thing about PubMed is this review tag. Okay, that is super useful. Okay, but if you happen to know that you know, if there's some good you know, two very good review articles are Trends in Cognitive Sciences, and then you have to just see what, you know, everything has a little abbreviation which may or may not make sense. So for instance, Trends in Cognitive Science is actually Trends in Cognitive Science, and you won't get hits if you miss off the n bit. But, you know, that's just trial and error bit, or you can read it straight from the results that come. Okay, so here's like, you know, here's some stuff, and this is actually one of the readings for like Rachel. This is a reading for Rachel Wu's talk later. Okay, so now you're getting somewhere. Okay, now if you say, okay, I want to find out about some area, it seems so overwhelming. Okay, well now at least I've actually got some, you know, any one of these is going to be, look, this one's 8 pages long, this one's 7 pages long. Okay, these are very manageable things to look at. Okay, and I would highly recommend taking these as initial jumping off points when this feeling of overwhelmingness hits you, because this feeling will hit you, probably. Okay. yeah, any one of these would be great. But another very good journal is Current Opinion Neurobiology, which also has a really weird PubMed abbreviation. But again, you can just get that. You can actually, there's a tool in here where you can look it up, but you can actually just get it just from sort of, seeing how things show up in results. So these would all be really good articles too. And in fact, Ben Hayden's talk, the reading for that is one from Current Opinion Neurobiology. And you'll see when you look at that paper eventually, that this does a really nice thing that it actually will, in the bibliography, it will highlight, the authors highlight nice papers with 1 or 2 dots, depending on how interesting they think they are, and put little mini summaries of the highlighted papers. Okay, so these journals really go out of their way to be helpful. Okay, oh, look at this one, here we are, the neural basis of smooth pursuit eye movement. So if you're really interested in that thing we were doing before, you can read that one. Okay, so that's my sort of PubMed tip. So review articles, find them with this, and do this Boolean stuff. And then a Google Scholar. Google Scholar, I just want to show you one checkbox. Have all of you used Google Scholar? Okay. Well, actually, I want to show you 2 things in Google Scholar. I'm overrunning a little bit. Okay. So suppose I'm interested in, say, I don't know, objects and concepts and cortex or something. Okay. Okay. So and suppose. I'm trying to bring out one of Brad's papers. I must be using the wrong term. Okay.
Connectivity or something.
Yeah, there you go. Okay. Okay.
These people are all very related, though.
Okay, well anyway, okay, so you bring one of these up, okay, and suppose I decide, okay, here we are, okay, well, suppose I find that I'm super interested in this paper, okay, suppose I say, oh wow, this paper looks like it's on exactly what I'm interested in, okay, I really am interested in the distributed cortical network for face perception. Oh, it came out in 2007. Okay, well all I have to do is look at all the subsequent papers that cite this, and then I'll know everything that's happened in work on distributed cortical network for face perception since 2007. And if I click on this, it's been cited 244 times. So all I have to do now is read these 244 papers and I'll be all set. Okay, that's a lot of papers. Okay, so this is the most useful checkbox in the whole of science, okay, in my opinion. Okay, this one, search within citing articles. Okay, so if you say, okay, I found my favorite paper, it's from a few years ago, I want to find out everything that's happened since then, but lots of stuff has happened since then, so I want to find, of the stuff that's happened since then that is in this area, there's some papers that are important enough that anyone who's working in this area will cite that paper. Okay, so find one of those, find some highly cited paper, preferably a review paper or something, and then say, okay, now what's gone in that, but I'm particularly interested in, say, patients with lesions, okay? So if I just type lesions in here and hit search, I'm just going to get a search for lesions. I'm not going to get a search that's related to that stuff that cited that paper that's about lesions. Okay, so I've lost it. Okay, now I've just got, now I've got papers from 1982 that are obviously very important papers, but don't cite that because it came out in 2007. Okay, so I hit this little search within citing articles box. Okay, now I'm going to get papers which are about lesions, which are in the general theme of, because they cite this paper about distributed cortical network for face perception, okay, and now I'm getting somewhere, right? Now I've got like a, more specific set of papers that are all pretty much on the kind of topic that I want, okay? And then I suppose, as I say, no, I want to find out, oh look, here's a recent Trends in Cognitive Sciences review on this topic, okay? So we're like, okay, I found a recent review paper on my topic, I read that, now at least I'm not, splashing around in a huge pool and drowning. Now at least I've got like a foothold. I mean, that's not the end of the story, but that's going to, you can look at the papers at that site, you can look at the papers at site that, so now you've got a handhold, okay? But even if you say, okay, well, the top hit turns out to be, they're sorted more or less by amount they've been cited, you say, okay, I want the really recent stuff, then you can go into here and you can say, what's happened since 2013, okay, and then you only get the more recent stuff. Okay? So this is a very, very good way if you say, okay, I'm interested in topic X, what's the very latest and greatest that's happened in topic X? This is a very good way of finding that. And I kind of particularly emphasizing this because I really did notice again and again in the projects that people were doing from last class, which are going to kind of be similar to this one, that people were like, I'm really interested in topic X, and they spent all this time thinking about it, and they totally failed to notice that a paper about exactly what they were talking about came out last year, and so everything that they were writing was a little bit redundant because of that. And then I would like Google Scholar for like 3 minutes, and I'd find the paper, and there's no magic to it. It's just basically using this little checkbox. So that's hopefully a useful trick. Yeah, you can also even like get Google Scholar to send you like alerts and alert you when stuff related to your own work happens, and that's really nice too. But we can talk about that some other time. So, so those are my. Yeah, I would say actually, being able to not feel like you're drowning in way too much stuff is probably, the most important thing, because especially when you're beginning graduate student, if it makes you feel any better, we all feel like we're drowning in too much stuff because there's just so much stuff, okay? But if you can search through it, then and find reviews, find things that are on the particular subtopic that you're interested in, then it starts to get a lot less overwhelming. Okay, I mean there'll still be quite a lot of stuff, but it's it's not 15,000 hits.